I will be able to guide you through how we use computer simulations here at HITS to solve some biological puzzles that we need for several applications. So let's start with the basics. This is a protein, and a protein folds like a puzzle. It folds itself. And actually, the term protein folding was coined in 1936, and it was inspired by origami. So that's why I gave you these sheets, and we're going to be doing some folding ourselves. And so I'm just going to say that we do a lot of protein folding and protein interactions. It doesn't mean that I'm actually good at origami, so bear with me on that. But let's see. Raise your hand if you ever solved a 3D puzzle or if you ever folded origami. Okay, lots of people have, so maybe you can help each other on that. But this particular protein is a synthene 1, and the name of it doesn't matter. What I want you to kind of imagine is that why is it folding that way? And it folds that way like an origami, much like an origami, to perform a certain function. In this case, for synthene 1, it regulates cell traffic. So it has that shape not by chance. It has that shape because it helps it do whatever it's supposed to do. So when we're simulating these proteins, we are looking at this, as Peter said, like in a video game, and just watching it and seeing what is it going to do. But if only it was that simple. Proteins are not by itself in the cell. So cells have thousands of different proteins, and they're all folding and interacting at the same time. So there's a lot of crowding, and here I'm showing some of it, but we can also add other types of molecules, not just proteins, such as genetic material, DNA, RNA. And if we zoom out a little bit, you can see that it gets really crowded in there. If we zoom out a little bit more, this is kind of the inside of the cell. And if we go on the outside of the cell, we can see that there's a lot going on there. There's this membrane, there are proteins in this membrane, and again, they all look different. So how can we study this with computer simulations and understand what these proteins are doing? Because if we want to, for example, build materials that resemble biological materials, which are often really, really good at doing what they are supposed to do, or if we want to test new drugs, or if we want to just understand disease or a particular process, we need to be able to capture the important bits of information that we have in this very, very crowded, very complex environment. And this is where computer simulations come in, because if we are just looking at this, you won't be able to see anything. And of course, there are tools like microscopes that you are able to see certain things, but this is a very complex environment, so it's very, very difficult to study a cellular environment in an experimental setting. But with computer simulations, we can choose what kind of things we are looking at, and we can model each of these particles individually. So it's like we are building a microscope that we can use and point wherever we want, and we can look at how much we want from the protein. But let's go back to our original protein. So again, this protein is folding in this specific way. So since we're talking about folding, maybe we can start with our origami. So get your sheets of paper. So each sheet of paper is going to represent an unfolded protein. An unfolded protein is composed of amino acids, which are just small building blocks to protein, and they look kind of like this. I'll pass these models around afterwards so that you can have a closer look. But they're very, very small bits of molecules that get together to build every single protein in our bodies and in every single organism. For the ones in our bodies, there are 20 different natural amino acids, so they combine in different ways to form proteins that do different things. So again, your sheet of paper is a chain of these bits, amino acids. Now let's start folding. So first thing we're going to do is fold the bottom point up to the top point. So you're holding your paper like this, and you can fold the bottom point up to the top point. And I'm going to go kind of fast here because I want to see how good you are at solving this origami. Now we're going to fold the right point over to the left point. And if you're unsure what to do, just do what feels right. If my instructions are not clear enough, just do what you think you should do. Now, I gave you a little help on this one because it's kind of difficult. Bring the flap back over and open it out at the same time. So you're going to do kind of what's shown there, like this. And then flatten the paper, aligning the top points. So as we're folding, we are getting into something a little more complex, right? Now we don't have just a flat sheet of paper. Now we have something that looks like something that's got a little structure already. And proteins do the same. So instead of having just a huge flat chain of amino acids, they fold into these building blocks that are a little more complex. And we call those alpha helices or beta sheets, for example, or regions that are a little more disordered, which we call loops. So you will see in my models of proteins later that there are certain parts that look very organized, like a coil, or something that looks like a flat bead, and this is kind of these intermediate structures. Okay, so now flip the whole paper over from left to right. And then bring the right side over to the left, opening up at the same time. So again, it's kind of the same as the previous one, but on the other side. So you're going to get at the end kind of like a square when you flatten the paper, aligning it with the points. Okay, so for those of you who didn't get that part, so you had something like, now I can't go back, something like this when you flipped it, and now you're going to do the same you did before. So you flip this one over and then fold it as a square. Okay. Okay, so next, fold the top point down to the bottom and then repeat on the back. And if you aren't following anymore, that's okay, don't worry. Okay? So now flip the right section over to the left. So you should have something like this at this point, and then you're gonna flip it like this. And you're gonna get the different color on the outside. And flip the paper over and repeat the last step. Okay, so you should have something that looks like that now. And again, if you don't, it's totally fine. That's the idea. I don't have it, so somehow I got lost on my own thing. Okay. Now you fold the left and right upper diagonal edges to the center of the crease. And then you will find number 12. Yeah, I already lost, so I'm just gonna use the one that I made before. Okay, so then flip the paper over, so around, and repeat the last step. And then fold the top point down to the bottom point. Okay. Give you a couple more seconds to figure it out. Now, unfold the previous step, so you should have something that you folded like this, and then you just unfold it. This was classified as easy on the website, so I just want to say we're all struggling, but you know. Now, flip the left and right sides of the model again so that you have bunny rabbit shape. So now you should have something that looks like a bunny rabbit. And again, I can't do it because I clearly missed a step. Okay, now fold one layer from the bottom upwards as far as it will go. So again, like you do the opening thing, if you had it like this, you do like that. Okay, and then now fold the top of that flap, you just fold it back down to meet its lower edge. So you would have something that you folded up, and then you just get the top and fold it again so that it meets the bottom. Now open the lower section and insert the flaps you've been making underneath it, hiding them completely. So you should have kind of like a pocket at the bottom, and you will fold that flap in. Okay? And then you repeat everything on the other side. Is anyone still doing it? Okay, so let's go. Now, fold the bottom section up. So this 21 is what you should have right now, and then you're folding that up. If you don't have that, something went wrong. Okay? Unfold that step. We're almost there. Now, flip the sides of the model on the left and right. So again, you're going to switch the colors. And now you will fold the top point down and then upwards. And you have a tiny overlap, so you can see over there. So you fold that down and then up, and then you have a little bit of a tip there. And then you fold that down to meet the bottom line. And then you fold that flap under the pocket. So that pocket over there, you're going to put everything in. Okay, and now you repeat the process on the other side, and you should find something like that. And so once you get to that point, you can start opening the box. It's going to feel a little awkward, but try to kind of flip it around and push the bottom of it. And that's going to be look kind of like this. So I did it before. Somehow I don't know what happened. But then you will find this. So a box. Did anyone get it? No. Oh, one person got it. Yay! Amazing. That's awesome. Was it hard?
I got it at home, Starbucks.
Ah.
But it's usually
Cool, yeah, that's nice. That's even better. That's more difficult. So you see, he said it wasn't hard. So I guess we're all kind of behind that point. But it took us a few minutes to try to fold this. And we didn't manage because there were so many different steps and there were so many ways that we could fold the paper. And proteins are exactly the same. They can fold it in many, many different ways, and the interactions that are happening within this amino acid chain are so intense, and in this crowded environment, it is amazing that they managed to find the exact right shape that they need to perform this function. I can put something inside this box. It's a box. Can you do the same with yours? Probably not, if you didn't build the box. So yours won't perform the function, right? And this is the same that happens with the protein. If the protein folds in a different way, or if it fails to fold, then we will have something that cannot do what it's supposed to do, and we will have a disease, or we will have inviable organism. So it's really, really important that our proteins fold the correct way. But while it took us a few minutes to try to fold this, the computer can fold thousands of the proteins that we are looking at in a second. So this is the advantage of working with computer simulations, because we can look at, even if we fold it in the wrong way, it doesn't matter, because we can try all of the ways. And we can do that several times, and eventually we will get the way that we're looking for our protein. So it allows us to explore all the possible folding pathways. And what happens is, every time that we made a fold, or every time that the protein folds, there's an energy cost, or to be more precise, an energy gain, because it's going to be more stable for the protein to be in its folded state than it is to be in this just chain of amino acids. So while we were spending energy, the protein is gaining energy, and some states are less energetic than others. And the state that's more favorable for the protein to be is the correct state, the state that it's found in nature. So we have here a plot, and we can see that the energy grows that way. So the completely unfolded protein, or our flat piece of paper, is the most energetic possible way. And then we can have, some folds that look like what they should be, some folds that don't look like what they should be, and they may all have very similar energies, but we want the protein to get where? Where do we want the protein to get? The lowest. So we want the protein to get around here, right? And that's the correct state. So if we find ourselves over here, that's not good, because we're going to get stuck. So that's what happens sometimes, and we get these misfolded shapes, and we can't really get the box anymore because we're stuck, right? Whereas if we do get the correct shape, we are now going in the right way, and then here we get something that doesn't look quite like a box, and here we get something that is performing the purpose of being a box. So it's the same thing with proteins. And we can do that with the computer and just try all of these ways to find which way the protein is going to fold. Now, if we want to understand physiological processes or biological processes, we don't want to just know how the protein folds. We want to be able to look at it interacting with all the stuff that I showed you before that it's inside the cell, right? And so we can do that also with simulations. It's kind of also very amazing, I think, that we do that by solving something that is quite fundamental, which are the equations of Newton. So here I have one of the laws of Newton's: force equals mass times acceleration. And this is what I do most of my days. Just solve this equation repeatedly and forever. Just sit on my chair and do that. Of course, the computer does it. But what we do is we start with the system that looks like this, and then we put a little force in it. So it's kind of like, imagine again our protein energy diagram, right? The well, and we have a ball at the top, and we want to know where it's going to end. What do? Just kick it a little bit, and then let it go, and it will find the lowest energy path. Same thing here. We kick it a little bit, we give it a little kick, and then we calculate the velocity of the protein, and with that, we can calculate the acceleration and calculate a new force. And we repeat that until the system completely forgets that we were kicking it in the first place, and it's now behaving as it would behave in nature. But what changes these forces? Well, again, the systems are not there by themselves. It's not just one atom in isolation. We have interactions, and these interactions change the trajectory of these particles. So we have the protein interacting with itself, we have it interacting with other proteins, we have it interacting with genetic material, we have it interacting with the medium, say water. I put some water molecules here. And all of these, we need to know how it happens. And of course, this is where we come in as researchers, because we're trying to figure out how these things interact so that we can model them in the most realistic way as possible to answer the questions that we're interested in. And so once we can do that, we're going to kick it, and then it's going to move a little bit. And here I'm just moving the waters. I'm cheating a little bit, but you get the idea. And what we get, it's something that's quite amazing. And I'm going to show you right now. But I want you to just look at this video that I'm going to show you with some appreciation for what is going on there. So every time that there's a little movement, that is thousands, millions of times that we've calculated the equations of Newton and moved these particles. So we can do that for our particles, and this is a video from an actual simulation from one of our collaborators in Japan, and I'll show you how they did that, or kind of what type of equipment they used to create this. But you can see that it is very chaotic, right? The proteins are interacting with everything that's around them, and they're not static, they're also moving and flapping around, and you have all these smaller molecules around as well, and each protein can have thousands of atoms. So for each protein, for each atom, we're calculating these forces. So you can imagine this can become very, very complex for the computer, even for the computer to do, right? And so you have this web of forces just being updated every time. The movement of these atoms is very, very fast. So they oscillate in the order of femtoseconds. And just so you have an idea of what that is, if I would get 1 second and stretch it so that 1 second is now the age of the universe, 1 femtosecond would be a third of a second, of a current second, not of the universe second. So it's really, really, really fast. And again, we have to be able to update that at that time scale so that we can watch what the protein is doing. But we also want to be able to watch it for a certain amount of time, because these processes in our body are not happening sometimes, or most of the times, in the order of femtoseconds, right? So we need to calculate this for a long time and many, many, many times repeatedly. And of course, the video that I showed you here was from a cell, and cells have billions of proteins and other molecules. So simulating an entire cell with everything that's in it, it's still not possible. People are still working on that. But it's the goal, kind of, for us to do as computational biophysicists. So here's the computer that I was telling you about from our collaborators. This is one computer in Japan, is one of the largest supercomputers in the world. And I told you that these proteins are moving, or the atoms are moving in the order of femtoseconds. This computer can do, I think, about 400 billion calculations per second. So then you can see how we get these calculations to work, because they're very, very powerful. But there are other things we can, sorry, not billion, quadrillion. Then it's even much, much more. So we can simulate these proteins for a longer time and watch them in nanoseconds or microseconds or milliseconds. Now, we can also be smart about it, because that's kind of what we're trying as scientists, right? So we're creating new algorithms that can help us look at particular parts of the molecule or particular levels depending on what questions we're asking. So we can, for example, use something like what I was just showing you, which is we call classical molecular dynamics, just the equations of Newton, to look at something that doesn't involve, for example, electronic states, doesn't involve any bonds breaking, doesn't involve any electrons, because electrons are particles that behave quantum and classical wise. So we need to also consider this quantum behavior, and then we would need something that is much more fine-grained. It takes much more math into account, but it takes much longer as well. So depending on the problem that we're looking at, we can get away with using several types of models. We can just treat everything as one equation. We don't even have to simulate atoms for certain cases, but we have to kind of be smart about it and figure out which one we can use depending on what we want to study. Another thing that we can do is simplify our systems. So instead of simulating perhaps every single atom in my protein, I can say that maybe each amino acid is one bead, if I know kind of how that amino acid interacts with the rest of the system. So we can have that kind of thing. I'm also going to pass this around. It's just some 3D models of proteins so that you can have a look. So there are a few things we can do to kind of deal with those limitations that we have in computer simulations. But, of course, what I want to say is, even if we're able to simulate this, our force calculations aren't perfect, right? Because we cannot possibly consider everything. Because again, we're ignoring electrons, we're ignoring certain interactions, we're ignoring perhaps the effect of anything that's inside the cell that we don't perhaps know. So we have to be very careful how we interpret this, because even though we call them computational experiments, we will always get a response, right? So we need to be very critical about our results to make sure that we're interpreting them correctly and we are making the claims that we can claim based on our models and based on our calculations. Okay, so let's wrap it up. So we started with a protein, or our box, our one box that one person could do it. And we saw that that was already difficult by itself, right? Now, we added a lot more proteins around it when we get the cell, and so it becomes a very, very hard system to solve and to watch and to look at. So I showed you that, I hope, simulations can help us solve these kind of problems and help us understand what's going on at the molecular level. So we can, for example, look at interactions between amino acids and how they react with each other, like I'm showing here. We can look at how proteins fold and what kind of the pathway that they take to get there. And we can also look at different levels of interactions. So for example, here I have the protein that's modeled, and again, like I'm showing you the loops and things. And then here I have something that's more precise, because that is maybe a drug that I want to know how it really reacts with the protein. So we can use different strategies to make this work at the level that we want. And every day, researchers like us are using these methods to look at real-world problems. So something like understanding COVID-19, understanding how we can produce drugs for it, or simply looking at metabolism, how the cell kind of maintains itself alive, how it mutates for a tumor. All of these kind of things can be done with molecular simulations, and of course now with machine learning, that opens up another point in that axis that I just showed you, right? Because you can then use data to kind of simulate these things, and you don't have to rely only on the physical parameters. So there's a lot still to do in this field, and I hope that I got you excited about it so that you can explore more and read more about it. And if you are excited, I just want to point out that most of what we do, although the pictures look very cool, is coding. So just know that we are doing all of this by, doing a lot of math and a lot of coding, which is all behind these amazing pictures that we can get now, which is by itself really cool. So thank you very much. I'm happy to take any questions, and I hope you enjoy the rest of the open day.